All right, welcome back everybody. Today we're going to continue through the endocrine system, and we're going to have 2, possibly 3 more lectures left in this chapter, and today we're going to begin studying the pituitary gland, which will really be a significant part of this chapter. So today we will introduce one part of the pituitary gland, and next lecture we will look at the other part of the pituitary gland, and we will see just how similar and how different the 2 portions of the pituitary gland are. So let's go ahead and get started with our not really attendance questions. First, most feedback loops in the body are of which type? And next, which structure sits within the sella turcica in the skull? Let's go ahead and try to answer those. Hopefully you've done that. And these go back to Bio 137. We have talked about feedback loops this semester, but the structure in the skull, that might take you way back. So first, what type of feedback loop is most of the feedback loops in the body? What type of feedback loop are we talking about when we talk about most of the feedback loops in the body? Most of them are negative feedback. There are a few positive feedbacks, but they're rare, and they tend to focus on things that are not constantly occurring in the body. So negative feedback loops, most feedback loops in the body. And which structure sits within the sella turcica in the skull? Remember the sella turcica? That means Turkish saddle. It's that little saddle-looking structure inside the cranial cavity. And that is where the pituitary gland sits, which is what we're going to be talking about today and next lecture. So here we're seeing kind of what the pituitary gland is. Up here is the full brain, and kind of hanging off the bottom of the brain is where we find the pituitary gland. And the pituitary gland is hanging right below a part of the brain called the hypothalamus. So the hypothalamus extends downward, and then we have the pituitary gland. Connecting the two, so in between the hypothalamus and the pituitary gland, we have this stalk feature right here called the infundibulum. The infundibulum. Now, when we look at the hypothalamus, it is a part of the brain. It is nervous tissue. So here we can see some neurons in the hypothalamus, and we can see the cell bodies, the soma. There's collections of them in the hypothalamus. And remember one of the terms that we learned back in Bio 137, a cluster of neuron cell bodies in the central nervous system is called a nucleus. So a nucleus is something that we're going to be talking about quite a bit. And nucleus, it's kind of unfortunate, but we have that word nucleus meaning a few different things in biology. But in this chapter, when we talk about a nucleus, we are meaning a cluster of neuron cell bodies in the central nervous system, specifically here in the hypothalamus. Now we will look at the specific nuclei as we go through the next couple of lectures. And we also see the axons extending from those neurons, and they are passing either completely through or at least part way through the infundibulum, that stalk. And in this image, we can see that some of those axons are extending all the way down into the pituitary gland. But now let's look at the pituitary gland itself. The pituitary gland is actually 2 different glands stuck together. Here on the right side of your screen, this kind of tan color is called the posterior pituitary gland. It's also known as the neurohypophysis. This word right here, neurohypophysis. Neuro because it's also made of nervous tissue. those axons that extended down from the hypothalamus make up the posterior pituitary gland. The whole pituitary gland is also sometimes just called the hypophysis. So neurohypophysis just means the part of the pituitary gland made of neural tissue. Now over here, on the other side, this is the anterior pituitary gland. And the anterior pituitary gland is sometimes called the adenohypophysis. Adeno means glandular. So the anterior pituitary gland, the adenohypophysis, is not made of neural tissue. It is glandular tissue. So right away we're seeing that the pituitary gland is two different glands. It's two different structures. One of them is a true gland. It is made of glandular tissue. One of them is not a true gland. It is made of neural tissue. And if we look even closer at this, the neural tissue in the posterior pituitary gland is the ends of the axons, axon terminals. Now, if you have not already, prior to starting this chapter, it is a really, really good time to go back and study how axon terminals work, because that is going to be very important for understanding how the posterior pituitary gland works. Now, today we are going to focus on the posterior pituitary. In our next lecture, we are going to focus on the anterior pituitary. But before we dive into that, let's actually take a look at how the pituitary gland forms. So here we have an early stage embryo. Here, 8 weeks, 16 weeks, but here at 4 weeks, before the pituitary gland really fully begins, what we're looking at is right here is going to eventually become the brain. And right down here at the bottom, kind of outlined in blue, this is an extension from what will become the hypothalamus. So this will become hypothalamus. This starts to extend downward. So another way to think about this is a piece of brain is extending downward. This will become the posterior pituitary gland. Now, at the same time, here is going to become the mouth, and there's some glandular tissue there. Salivary glands will eventually sprout from this. And some of what will become salivary glands kind of take a different detour. They start to pinch off in a different direction. Instead of extending to the sides of the mouth and under the tongue, they start to extend upward towards the brain. And down here now, as we go a little bit further, we can see that a piece of that, what would have become a salivary gland, breaks off, starts to migrate upward. Meanwhile, that neural tissue is extending downward. They bump into each other, and that glandular, almost salivary gland eventually becomes the anterior pituitary. That neural tissue from the brain extends downward and becomes the posterior pituitary. So they truly are two completely different glands, different origin, different structure, different functions. Now, like I said, we are going to focus on the posterior pituitary gland. So let's ignore the anterior for now. Looking up here again in the hypothalamus, we are going to look at two of those nuclei that we just mentioned. The first is higher up in the hypothalamus. This is called the paraventricular nucleus. Para means near or around. Ventricular, the ventricles of the brain. Remember those hollow spots that were filled with cerebrospinal fluid? So this nucleus is higher up near some of the ventricles. We also have something called the supraoptic nucleus. Supraoptic nucleus. Remember the optic chiasma, where the two optic nerves cross? Well, just above that, just above the optic chiasma, we have the supraoptic nucleus. Now, from the paraventricular nucleus, we have axons extending down through the infundibulum, and we have the axon terminals here in the posterior pituitary. From the supraoptic nucleus, we have axons extending down through the infundibulum and ending here in the posterior pituitary. Now, let's think back to Bio 137. What do you remember about axon terminals? What is stored there? Hopefully you remember that stored in axon terminals are neurotransmitters. Where were the neurotransmitters made? They were made in the soma of the neuron. So putting all of that together, these neuron soma up here in these nuclei are making neurotransmitters. Those neurotransmitters travel down the axons and they are stored in the axon terminals. When are neurotransmitters in axon terminals released? When an action potential passes down the axon, it causes the release of neurotransmitters. Now, in this case, these neurotransmitters are also hormones. We're going to be looking at two hormones today. Cells up here in the paraventricular nucleus make a hormone called oxytocin, abbreviated OT. Cells in the supraoptic nucleus make a hormone called the antidiuretic hormone. Sometimes it's also called vasopressin. Antidiuretic hormone, abbreviated ADH. So, oxytocin is made in the somas of the paraventricular nucleus. It travels down the axon and it is stored in the axon terminals of the posterior pituitary. ADH is made in the soma of the supraoptic nucleus, travels down the axons, and it is stored in the axon terminals here in the posterior pituitary. Now, if I ask you, where is ADH made? Well, the answer to that is it is made in the supraoptic nucleus of the hypothalamus. If I ask you, where is ADH released from, or which gland releases ADH, you would say the posterior pituitary. Now, that's the case for oxytocin also, except it's the paraventricular nucleus. ADH and oxytocin are unique. Every other hormone in the body is made by the same gland that releases it. But ADH and oxytocin are made by the hypothalamus. They are released from the posterior pituitary. No other hormones follow that pattern. All other hormones are made by the same gland that release them. We're going to come back and look more at oxytocin and ADH here in just a moment. But now let's look at how do these hormones get into the blood? Well, again, they're made up here in the hypothalamus. They travel down and they are stored in these axon terminals. When an action potential comes down, those ADH or oxytocin, depending on which ones are sending the action potentials, ADH or oxytocin, are released from those axon terminals. It is surrounded by a capillary bed. Those hormones, ADH or oxytocin, move into the capillary bed and travel off to the rest of the body. So let's look at the path of blood when we are talking about pituitary hormones, specifically posterior pituitary. Blood is traveling from the heart through a large vessel called the aorta. If it's going towards the brain, then from the aorta it moves into the common carotid artery. The common carotid artery is a blood vessel that takes blood to the brain. Now, the common carotid artery splits into an internal and an external carotid artery. If we're going to the pituitary, then we go from the aorta to the common carotid artery to the internal carotid artery. One of the branches that comes off of the internal carotid artery is called the inferior hypophyseal artery. That then takes us through this capillary bed that surrounds the posterior pituitary. This is where those hormones, ADH or oxytocin, move into the bloodstream. Once that blood picks up either ADH or oxytocin, it's going to leave the posterior pituitary through the hypophyseal veins. It enters a larger vessel called the cavernous sinus. That is a vein. And then it joins the internal jugular vein. The jugular vein is the vein that drains blood away from the brain. From the jugular vein, the internal jugular vein, we join the brachiocephalic vein into the superior vena cava, which is the blood vessel that returns blood to the heart. So, know this path of blood for the posterior pituitary. I will tell you, every single exam, we are going to have a question where you must tell me the path of blood from some point to some point. Listen to what I just said. I'm telling you a question that will be on every exam. Every single exam will have a question about the path of blood from some point to some point. This is the path of blood for posterior pituitary gland hormones. All right, so here we are again, back with ADH and oxytocin. Right now, we are going to focus on ADH. ADH made in the supraoptic nucleus, travels down these axons and it's stored in the posterior pituitary gland. So let's take a look at ADH. ADH, I said, also called vasopressin. Those two things are the same thing. Antidiuretic hormone, vasopressin, they're the same thing. There's just two different names. It is a peptide, so it's amino acid based. It's made in the supraoptic nucleus. It's stimuli. What causes it to be released? Primarily, the number one stimulus for ADH is increased plasma osmolarity. increased plasma osmolarity. When does increased plasma osmolarity occur? When we are dehydrated. ADH is the hormone that keeps our body hydrated. When we have increased plasma osmolarity, ADH is released. But there are some secondary stimuli. For example, low blood volume. If our blood volume is low, ADH is released. Low blood pressure. If our blood pressure is low, ADH is released. Pain and some drugs also cause ADH to be released. But again, the primary stimulus for ADH is increased plasma osmolarity. Now, we are going to talk about ADH all semester long. So, every time we talk about it, it is almost always going to come back to increased plasma osmolarity. Keep that in mind. It is inhibited by adequate hydration. If you have plenty to drink, then you are not going to release ADH because you are well hydrated. And really the job of ADH is to make sure you are hydrated. If you are, we don't need to release it. It is also inhibited by alcohol. Now, keep that in mind. We're going to come back to it in just a moment after we talk about the function of ADH. How does it work? Well, at the kidneys, we have these tubules called nephrons. The functional part of the kidney that we will study later this semester is called a nephron. And there's these little tubes that make up the nephron. Near the end of those tubes, there's something called the DCT and the CD. You don't need to worry about those right now. Just know that these are small little tubes inside your kidneys. ADH tells those tubes to create aquaporins. What are aquaporins? We mentioned them in Bio 137 early on. Aquaporins are tunnels in your cells that allow water to pass through. So if there are more aquaporins, then more water will pass through. What do we mean by pass through? Well, here your kidneys are removing water from your blood and it will eventually become urine. But if you are dehydrated, we don't really want to lose a lot of water in the urine. Instead, ADH tells your kidneys to make aquaporins so that water can go back into the blood. Instead of leaving the body as urine, it goes back into the blood. That is called reabsorption. Reabsorption. We absorbed it once when we drank it, but now we are absorbing it again at the kidney so it does not leave the body. ADH tells your kidneys to make aquaporins to reabsorb water. That way we stay hydrated. That water is reabsorbed, it goes back into the blood, so our blood volume is increased. And that ultimately increases blood pressure. If we have more blood, there's going to be more blood pressure. Another thing that ADH does, we said it's also called vasopressin. Well, vaso means blood vessels. So vasopressin tells us that it has this other job related to blood vessels. It causes vasoconstriction, blood vessel constriction. If our blood vessels are constricting, it means they're squeezing harder on the blood. Well, that also increases blood pressure. So ADH keeps us hydrated and it keeps our blood pressure elevated. Now, let's go back to this alcohol thing. I do not drink, but I have been around plenty of people who do, and whether you drink or not, you know what happens when someone drinks alcohol. What happens? They go pee a lot. Way more than the amount of liquid that they're drinking. It's not that alcohol is just passing through them. No. What's happening is alcohol is inhibiting ADH. If ADH is not being released or it's not having a function, what happens? Well, we do not get aquaporin production. We do not increase water reabsorption. So instead, more water leaves the body in the urine. ADH does not have a function when you are drinking alcohol. The reason that someone who drinks alcohol pees a lot is because it blocks ADH. All right, so let's talk about hypersecretion and hyposecretion. Hypersecretion of ADH leads to something called SIADH, which is the syndrome of inappropriate ADH production. Too much ADH. And if you have too much ADH, well, all that happens is these functions up here are elevated. So you increase your blood volume, you increase your blood pressure a lot. And that can cause high blood pressure, hypertension, because of elevated blood volume. So SIADH, syndrome of inappropriate ADH production. But what if we go the other direction? What if we have hyposecretion? Now here is where we start to really get into some different types of diabetes. When we were talking about the pancreas, we said that there's a lot of different types of diabetes. And some of them have nothing to do with insulin, some of them have nothing to do with glucose, and here is our example. Here we have something called diabetes insipidus. Diabetes insipidus. Now there are two types of diabetes insipidus, but both of them have these things in common. Polyuria, remember what that meant? Peeing a lot. Low blood pressure. Why would that be? Because you're urinating a lot, losing a lot of water, losing blood volume, lowering your blood pressure. The urine that comes out has a very low specific gravity. That just means that it is almost entirely water. Why is that? There is no solutes there to begin with, but more importantly, there's no glucose there. At no point did we say anything about glucose. That's because someone with diabetes insipidus has no problems with blood glucose. Their blood glucose is normal, therefore blood glucose is not going to spill over and cause glucose to be in the pre-urine. Instead, it's just a lot of water in the urine, and that water is not being reabsorbed because of ADH not being active, and as a result, the urine that comes out is mostly water. So let's look at the two types of diabetes insipidus. First, there is neurogenic, and second, there is nephrogenic. Neuro means nerve related. Nephro means kidney related. So neurogenic, in this case, ADH is either not being produced by the hypothalamus or it's not being secreted from the posterior pituitary. This usually comes from something like a blow to the head or a brain tumor near your hypothalamus or your pituitary gland. But either you are not making enough ADH to release, or it's being made in very low amounts, or it's just not being released. The other type is nephrogenic. This is usually genetic. It can be an autoimmune disease where the kidney tubules are attacked. But in this case, ADH is being made just fine. It's being released just fine. But when it gets to the kidneys, there is no response. The kidneys do not make those aquaporins. In both of those cases, the result is the same. You're not reabsorbing that water. It's leaving the body as the urine. And the urine is extremely, extremely dilute. It is almost pure water. So, let's take a break for a moment and learn a little something interesting. We're going to talk about diabetes mellitus. Remember, pancreas, insulin, glucose, and diabetes insipidus. Hypothalamus, pituitary, ADH, no glucose. What we've learned so far in both of those types of diabetes, well, even including, we also mentioned steroid diabetes. Lots of different causes, lots of different underlying factors. But there's one thing that all diabetes has in common, and that is increased urination, polyuria. So polyuria is what diabetes is about. It's not about blood sugar. It's not about insulin. It's about peeing a lot. But let's say, a long time ago, before modern medicine, before lots of current medical devices were invented, or even today in some countries, you wake up and you realize, man, lately I have really been peeing a lot and I need to go to the doctor. So you go to the doctor and you tell the doctor, I've been peeing an abnormal amount, and the doctor says, well, it sounds like diabetes. But there's a bunch of different types of diabetes, so we need to know exactly what type of diabetes you have. Maybe you have diabetes mellitus, maybe you have diabetes insipidus. Let's narrow it down. How do you narrow it down when you don't have machines and things to look at urine contents? Well, you do the old finger test. You dip your finger into a urine specimen sample and you touch it to your tongue. And when you touch it to your tongue, one of two results happen. It's either going to have a lot of glucose in it if they have diabetes mellitus, so it's going to taste sweet, like honey. Mel means honey. And when you taste urine from someone who has diabetes mellitus, it's going to be very, very sweet because of all of that glucose that's in it. Instead, if you dip and taste and there is practically no taste at all because it is almost entirely water, well, that's diabetes insipidus. Insipid means tasteless. It's tasteless because it is almost entirely water. So diabetes mellitus, glucose, honey-like. Diabetes insipidus, insipid, water-like. Aren't you glad for modern medicine? All right, so here's our feedback loop for ADH. We have normal water level in the blood, but if we start to get a little bit dehydrated, water levels drop below the normal range, your hypothalamus detects that. It's got osmoreceptor, so it is looking to see the solutes in the blood. Are they a little bit too high, a little bit too low? If they're a little bit too high because your water level dropped, Well, two things happen. One, your posterior pituitary is going to release more ADH. The supraoptic neurons send action potentials, and then those axon terminals in the posterior pituitary release ADH into the blood. At the same time, it's going to cause you to feel thirsty. Your hypothalamus is what causes you to feel thirsty. You're going to drink more. Your kidneys, because of ADH, are going to reabsorb more water. Those things bring your water level back up. Once your water level is up high enough, well, now your hypothalamus is going to detect low solute concentration. It's going to stop sending those action potentials, so the posterior pituitary stops releasing ADH. Your kidneys reabsorb less water, more water leaves in the urine, water levels drop back down to normal. We're constantly going through these two sides. The result is homeostasis. Water levels in the blood stay pretty well normal. All right, so now looking at the paraventricular nucleus in the hypothalamus where oxytocin, OT, is made. Oxytocin is also a peptide. It's an amino acid-based hormone. Its stimuli are cervical stretching, so baby about to be born, suckling, baby nursing at the breast, and intimate touch, whether this is holding your baby, whether this is cuddling with your partner, whether this is sexy time, any type of intimate touch also releases oxytocin. It is inhibited by cessation of those stimuli. So cervical stretching stops, baby's been delivered. Suckling stops, baby is full. Sexy time is over, or you stop holding hands, or you lay the baby down. All of those things stop releasing oxytocin. So what's the function of oxytocin? It stimulates uterine contractions. So those contractions of labor, that's the result of oxytocin. Milk ejection, so the release of milk from the breast. And love. So all of those good feelings that come from being in love with your partner, or loving your family, loving the baby, all of that we call love is oxytocin binding to nerves around your body. So let's take a look at this feedback loop. Here, the head of the baby is ready, it's in position, and time to start pushing, start delivery. The head of the baby pushes against the cervix. And when that happens, it's going to stretch the cervix. The cervix sends electrical impulses through the nerves to the brain. The brain, specifically the hypothalamus, receives those signals and releases oxytocin from the posterior pituitary gland. Oxytocin now traveling in the blood until it gets to the uterus. Oxytocin causes the muscles of the uterus to contract, pushing baby downward towards the cervix, stretching it more, causing more signals, causing more oxytocin to release, causing more contractions, causing more stretching, causing more signals, causing more oxytocin to be released over and over and over again until the baby is born, until delivery has stopped. Now, the big question: what type of feedback loop is that? That is one of the rare positive feedback loops. If we look back at our common feedback loop, what happens? Water level drops, we respond by raising water level. Water level is up to normal. What happens? We respond by lowering water level. So the stimulus is in the opposite direction of the response. But in a positive feedback loop, the stimulus and the response are the same. Cervical stretching causes more cervical stretching, and that keeps happening over and over and over again until the baby is born. That is a positive feedback loop. Similar, if we just look at the oxytocin part here, we'll talk about prolactin in our next lecture, but if we just look at the oxytocin part, what happens? Baby is nursing. That stimulates the release of oxytocin, which causes more milk to come out, which causes baby to nurse more, which causes more oxytocin, which causes more milk to come out, which causes baby to nurse more, and that keeps going until baby is full and unlatches. So again, positive feedback. Now the real kicker is, think about a partner that you've had that you were really in love with. When you're with them, you feel so good. And when they're not around, that feeling kind of doesn't feel so good anymore. So what happens? You go back to them to get more oxytocin. The more you're with them, the more oxytocin is released, the more you want to be with them. If you've ever had that feeling of being addicted to the person that you were with, that's oxytocin. That's why you want to be with them so much. And that's kind of intentional. That's kind of a positive thing, because that drives you to stay with the person, possibly driving reproduction. It all ties back into positive feedback. So everything we've talked about today are the hypothalamus, the supraoptic and the paraventricular nucleus, the infundibulum, and the posterior pituitary. Next time, we are going to talk about the anterior pituitary gland, and it is a completely different story. It behaves differently. Everything about it is different. So, if you have any questions at all, make sure to reach out. Otherwise, I will talk to you next lecture.